Well, good afternoon. I expect people to filter in as they get done networking and everything else, but the one thing that was really stressed is to be adamant about starting in time. So, I've already lost that.
Yeah, 5 minutes is gone, right? I'll catch us up.
That's awesome. So our first speaker in this research session is Craig Just. He's a professor at the University of Iowa. We won't hold that against him here in Cyclone
Yes, you
Country. He's going to give a talk about a study that utilized targeted and non-targeted 116S rRNA sequencing to determine how anammox bacteria and other in-cycle microorganisms differ in sediments with mussels compared to sediments with no mussels.
Yeah, who would ever submit an abstract like that, right? Wow, after dinner, or after lunch, you're trying to take this all in. Yeah, I know. Anyway, thank you for the introduction. I appreciate it. It's a pleasure to be here. I actually did go to Iowa State University for 1 year, but it was too hard. So then I went to the University of Northern Iowa. I got 2 degrees in chemistry there. And then I wanted to get my PhD and decided ISU would be too hard. And you and I didn't offer it, so I went to Iowa to the easy place. Got my PhD down there several years ago. Born and bred Iowan. I'm happy to be here. And I'm glad you guys are all here too right after lunch. Lots of people to thank, by the way. Students do all the work when you're a professor. And so I'm just here to share the good stuff that they've been doing for a long, long time. So I'm going to talk about 3 big picture research questions today. One will be, can the influence of native freshwater mussels on the nitrogen cycle be quantified, right? So nitrogen cycle in Iowa, kind of a big deal. Can mussels do anything about that? And can we prove that they can do that quantitatively? How might anaerobic ammonia oxidizing bacteria, I'll call them anammox from here on out, impact our nitrogen pollution legacy? And then lastly, how do mussels influence what's known as the nitritation anammox process in natural environments, all right? So a lot of you, this may not be your scientist, trust me, I'll lead you through it, all right? I will not talk over anybody today. So as you might know, managing the nitrogen cycle, it's been identified in my field of engineering as a grand challenge by the National Academies of Engineering. Over 20% of the land-applied nitrogen fertilizer in the U.S. Corn Belt ends up in the Mississippi River. Fertilization of the Gulf leads to harmful algal blooms, creating a dead zone that can be up to 7,000 square miles, stretches all along the southern coastlines of Louisiana and Texas into the shores of northern Mexico. Estimated dead zone losses to the $650 million Gulf fishing industry is about $20 million each and every year. Over 150 coastal dead zones of similar magnitude currently exist worldwide. And in Iowa, over 300 million pounds of nitrogen to the tune of about $150 million are lost each and every year. So that's our challenge. Native freshwater mussels are prolific ecosystem engineers that strongly influence the nitrogen cycle at the water-sediment interface. They are benthic burrowing, suspension-feeding bivalves, right? So they eat some of the products formed by over-nutrifying, essentially putting too many nutrients in our waterways. They filter these things, they eat that. They can be large and long-lived. Some of the pocket book mussels that I work with are as big as my extended hand, and they could be over 50 years old. They transfer nutrients from the overlying water to the sediments. They stimulate biomass production across multiple trophic levels. The biomass of healthy mussel beds, they kind of live in communities together, supporting each other in some ways, exceed that of all river bottom organisms by an order of magnitude. So as an environmental engineer, we want to work with organisms that are kind of big players in these ecosystems. Niche players are great too, right? That's what makes it an ecosystem. But if you're an environmental engineer like me that wants to address this grand challenge, we got to have big players in the system, organisms that really have a big influence on it. Annual mussel biomass production can equal that of all other river bottom organisms. And so, as I mentioned, they have a large influence on these geochemical cycles in the waters they inhabit. One mature Lampsilus cardia mussel can filter up to 10 gallons of water per day. They have an in-current siphon shown here where complex organic nitrogen in the overlying water is removed via filter feeding after it enters the in-current siphon. Then the excurrent siphon, they essentially exhale their water back out. But along the way then, they digest and make simplified reduced organic nitrogen and inorganic N that ends up being deposited into the sediments and end up being handed off to other microorganisms then that then can complete the nitrogen cycle, hopefully in our favor. I click, there it goes. So here's what it looks like for a freshwater mussel in one of my habitats that I just took down in my lab. I'd used it for several years and now it's gone. I'm going to miss it forever, but you can see a mussel here on the upper left. The in-current siphon sucking water in, the ex-current siphon pushing water back out, right? So an organism this size, as I mentioned, can filter 10 gallons of water each and every day. So one of our primary questions, can the influence of native freshwater mussels on the N cycle be quantified? We did a study with 25 Amblema plicata, known commonly as a three-ridge mussel, and then Lampsilus cardium, as I mentioned, is a pocketbook mussel. They're commingled in two mesocosm tanks. The reason, again, why we chose them is collectively they represent 40% and 30% of the mussels in the upper Mississippi River. In Pool 5, way up here, there might be 190 million mussels in a short stretch in that pool. Pool 6, 61 known mussels quantified here. Down here in the Pool 18 area, over 200 million mussels. So collectively, there are billions of mussels, some of them filtering 10 gallons of water per day each. They're doing a lot of filtration for us for sure. In fact, collectively filter 2 billion gallons of water per day during average flow in the Mississippi River. All right, so it's a big deal. So we did a 72-day experiment that I'll show you today. 65 days of mussel, acclimation and mesocosm ripening, that we call it. And then we had 2 no mussel control mesocosms in addition to the 2 that had mussels. We had a head tank that had Iowa River water, providing about 55 liters per water with a hydraulic retention time in each tank of about 2.5 hours. We had a submersible pump that kept the phytoplankton, as a generic term, suspended. Phytoplankton could be algae, could be other things, just green organisms that the mussels will eat. We had artificial solar irradiance on a 12-hour on, 12-hour off cycle. And then we did some intensive water chemistry sampling during the last 7 days of the 72-day experiment. We collected some water samples by hand in the overlying and in the pore water. And then, we also had some chemistry sensors that took measurements every 30 minutes. This is what the system looks like. Again, head tank with Iowa River water, 4 different tanks altogether, artificial sunlight, artificial lab person. We have some tanks here. Again, keeping the particulates in the water column to some degree. Mussels in the sand. We had a photosensor at the bottom near the mussels. We had a photosensor at the top. And then we have this hydrolab sensor that can do chlorophyll A, nitrate, ammonium, dissolved oxygen, pH, connectivity, all on 30-minute time intervals. This is what the system looks like when it's up and running. Yum, Iowa River water at its finest, green and murky and nasty. But nonetheless, that is the environment that we've created for many of our freshwater mussels here in Iowa. And so that's what we were looking to simulate in the lab. So phytoplankton decreased and all other nitrogen species increased in the presence of mussels. Duh, phytoplankton is their food, right? So in the presence of mussels, phytoplankton goes down by almost 50% compared to tanks that don't have any mussels. So they're eating, that makes sense. And everything else goes up. Ammonia, so I like to tell people that I spend a lot of money to study how freshwater mussels eat, poop, puke, and pee with respect to nitrogen. So mussels pee, right? Here we go. Ammonia, you and I will do that shortly as well from our lunchtime things that we're eating and digesting. By the way, I do know how long it takes mussels to digest their food. And so ammonia going up by almost 100% in the pore water. When we have sensors measuring the data, when we take samples by hand, it came out to about 66%. And then this is in the overlying water. In the pore water, it goes up about 160%. Nitrate goes up as well. Why would nitrate go up? Well, ammonia is a substrate for bacteria that then convert it to nitrate. So more ammonia in a relatively well oxygenated tank means you're going to get more nitrate. Using the sensor data, we're actually able to show that there's statistically more nitrate in the presence of mussels than without. And then last but not least, nitrite, also higher in the presence of mussels. and also total nitrogen higher in the presence of mussels, and also organic carbon higher in the presence of mussels. So mussel excretion products include organic carbon as well, right? And so they're adding organic carbon to the sediment as they do their digesting and excreting. We also made a numerical model so we could predict important water chemistry dynamics. I got 2 degrees in chemistry from the University of Northern Iowa, 1 degree in engineering from Iowa, and the main difference I can tell is that engineers like to make numbers do more things. They use math more than chemists. And so here I took my chemistry and my math, put them together to make a model that can predict what mussels do. Why do I want to predict what mussels can do? Because if I want to advocate to spend dollars to rejuvenate their habitat in the name of getting water quality benefits, I've got to be able to predict their impact and make a case to someone, hey, let's build some mussel habitat so we can get various impacts under various scenarios. And so we had all this model input, so temperature goes up and down each and every day. Nitrate, we had some nitrate spikes, boy, not surprising, in Iowa river water. And then we've got food, collectively, phytoplankton going up and down during time. We've got some ammonia inputs, and we've got nitrite inputs. So everywhere you see blue dots here, that's sensor data taken every 30 minutes. This particular panel is for food or phytoplankton. And then the line that you see down here is my computer model, and it matches pretty well. That's really the take-home message. If you look at nitrite data, I don't have as much nitrite data. Orange dots are nitrite. You know, a little hard to know with that signal whether or not we're matching that well, but it's also very low concentrations. Looking at ammonia, again, green dots are sensor data. Black lines are my model. And then last but not least, nitrate. Red dots are sensor data, and the black line is my model. So we're able to relatively, predict pretty well what's going on in the presence of mussels in these tanks as they eat and excrete. And so as a result of this, we are able to then use our model simulations also to predict mortality thresholds, because if you have high ammonia in the pore water, it's essentially the mussels not being able to get away from their own waste, and it becomes acutely toxic to themselves. And so that is a function of, again, food concentration down here on the x-axis, or as a function of total nitrogen. They're clearly related. And then mussel biomass too. So what that means is the more food you have and the more mussels you have, the more acutely toxic your environment becomes. Right? More mussels excreting in the presence of more food, going to be more toxic. And so we ran several models, and then we're able to simulate pretty quickly that if you have a high mussel density, around 500 grams per meter squared, pretty acutely toxic even at low food concentrations. Alternatively, if you only have like 50 mussels or 50 grams of mussels per square meter, it really doesn't become acutely toxic at really any level of food concentration. And actually this says unpublished data. It's actually just got published in January. I forgot to update this slide. But ironically, here I am looking for acute toxicity of ammonia on mussels. But the thing that's most acutely toxic to them is when we used to make buttons out of them. And so you can actually go to Muscatine, Iowa to the Pearl Button Museum or the Mussel Museum. I forget the exact name of it. But we used to harvest these mussels. We over-harvested them about 100 years ago almost to the point of extinction. And they've yet to even come back. So of all those mussels that I told you about that are in the upper Mississippi rivers, billions of them still, it's probably a tenth of what it was 100 years ago. And so again, the ecosystem in the past has had many more mussels that could give us those water chemistry benefits if someone like me can show up and advocate on their behalf. And so I'm trying, all right? I'm trying. So can the influence of native freshwater mussels on the end cycle be quantified? The concentrations of all major end cycle species were significantly altered in the overlying water of mesocosms with mussels, and the amounts were readily quantifiable. I could do it. Students could do it, candidly. A numerical model adequately predicted the aquatic end cycle dynamics with mussels at this scale. Mussel mortality was simulated as a function of food, essentially, mussel biomass and pore water ammonia. And the quantified mussel influences from this study were limited by the small spatial scale. And mussels significantly increased overlying water nitrate concentrations at the mesocosm scale. And so I ask you, mussels to the rescue? Nitrate is the most mobile form of nitrogen we can put in our rivers and streams. And if mussels are depositing ammonia to the river sediment, and then that gets quickly oxidized to nitrate, we might actually be using mussels to mobilize more nitrate. More to this story to come. Got to keep moving here, man. I got to gain 5 minutes back. As you know, Iowa's nitrogen-rich rivers are feed sources for urban drinking water. And so here's some data in the Raccoon River watershed, which we just heard of lately, where we're having maxima up to 18.5 milligram per liter nitrogen as a result of nitrate contamination, which is well above the drinking water standard of 10. And so the Des Moines Waterworks, as you might know, right down here in the Des Moines area, a pretty controversial lawsuit suing these 3 counties. I was born in Sac County, that's one of these 3. Sac County, Iowa, at the Raccoon River is about a mile from where I was born, at the Loring Hospital in Sac County. These, as you might know, these drainage districts were sued as a part of the lawsuit. And what we find out in the end is that tile outlets that look like this are not point source discharges. That's essentially what the courts have said. And so therefore you cannot regulate this non-point source. I'm sure it looks kind of like a point to me, but you cannot regulate that under the National Pollution Discharge Elimination System. So, Iowa's pollution legacy with respect to nitrogen remains to be seen how we deal with it, given that something as drastic as a lawsuit really couldn't force us to change our ways. So it'll be very interesting to see what we do in regard to that. So what got us into this nitrate problem in the first place, right? We can't deny that we have a nitrate issue. No one can deny that. No one is denying that. Farm Bureau, you name it. No one is denying that. We're grappling with how we're going to deal with it, which is fine. That was not the conversation just a few years ago. A few years ago, we didn't have a nitrate problem. We did then, but we certainly do now, and now we're just trying to figure out what are we going to do about it. The reason why we got into this issue in the first place is our ability to take nitrogen from the atmosphere via the Haber-Bosch process, turn it into ammonia fertilizer, put it on the land. These oxidizing bacteria, AOAs and AOBs, oxidizing bacteria and oxidizing archaea, are very good at converting ammonia to nitrate or nitrite, and then these nitrite oxidizing bacteria are very good at making nitrate. They're fast. They work really well. The ones that convert it back from nitrate to nitrite back to nitrogen gas, they're slow. They don't work anywhere near as fast as the others. So our ability to produce nitrate has been unbelievably influenced by humans here on the planet, and that's really got us in the predicament that we're in. All right? So, classic, by the way, I'm a wastewater guy. We deal with wastewater issues throughout the state. Oh, is that Ted? Speaking of wastewater, there's one of my wastewater guys, Ted, in the back. We take your ammonia that you flush down to our wastewater treatment plants. We do convert it to nitrite and then nitrate by adding a bunch of oxygen. And then we turn the oxygen off and turn it back to nitrogen gas. That's one of the ways that our urban centers do not put nitrogen into our rivers and streams. So we actually let it bubble away as nitrogen gas. Works really well. But one of the things that you can do in our more modern wastewater systems is to get rid of the step where you make nitrate over here. You just go ahead and oxidize to nitrite and have these new bacteria, relatively new, to engineers at least. Mother Nature's had them for a very long time. Where we can actually convert some of this ammonia and nitrite together to make N2 gas, bypassing that traditional way that we've gotten rid of nitrogen in our wastewater streams in the past. And the bacteria that do this are these beautiful little pink organisms called these anammox bacteria and archaea. So they definitely are impacting our nitrogen pollution legacy. They were discovered in '95. But it was actually predicted by a really smart person way back in 1977. It's been called the missing lithotroph in a 1999 article in Nature, really kind of completing our understanding of the nitrogen cycle. And you can find these planktomycetes organisms in the Black Sea, at the desert, in an estuary, and gorilla feces, right? First of all, amazing they're there, but who decided to look? I don't know. Somebody thought they should look. And current estimates attribute 50% of the nitrogen gas released to the atmosphere, over 100 gigatons per year globally, to anammox metabolism. So they are also a big player. Mussels are big players. Anammox organisms are big players. Very interesting to me as someone who wants to engineer such things. In the interest of time, I'm going to move by this. Message of this is, if you use anammox in your wastewater treatment plant, you use less oxygen, you save money. That's what that slide says. All right, so how do mussels influence nitrotation anammox in natural environments? So what we did is went and took some sediment cores from the Mississippi River and learned or tried to study ammonium excretion by mussels, and learned, if you kind of think about it, they could elevate nitrite. In fact, in the lab we have data that suggests, that actually showed they do elevate nitrate in these zones, but it can happen in very specific redox zones. So if you look at the concentration of certain things in a sediment column as you go from top to bottom, if you look at oxygen, right, it's oxygenated at the top, you go down deeper, oxygen goes away. If you look at nitrate, nitrate might be lower, although in Iowa, nitrate could be over here in the river water. But a lot of places it starts there, it gets oxidized by bacteria, and then goes back down over time. And that happens as a function of ammonia, right, where it might have low ammonia, but go down, 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 you're going to get more. And so then you can imagine this zone, right, this specific zone then where you might get a little bit of nitrite as ammonia is still being oxidized in the presence of oxygen right there. You can make a little bit of nitrite. Well, remember that ammonia and nitrite are influenced by mussels, right? They go up in the presence of mussels, and those just happen to be the substrates for this anammox bacteria that then produces nitrogen gas from those two substrates. So you can see how mussels might create the special zone where anammox bacteria might like to be. And that's what we wanted to study. And so the way we studied that, we used a non-targeted microbial community analysis technique in sediments with mussels and in sediment cores without mussels. We then took soil out at various depths. We extracted the DNA, and then we did what's called Illumina 16S amplicon sequencing to essentially sequence all the DNA in the samples and therefore identify all the organisms that would be in that sample. So if you look at a case where you've got no mussels and with mussels, and you look at the phylum level, they look about the same. Looking again at the order level, they look about the same. Each of these colors represents a different organism that we found. And then if you look at the genus level, you know, it looks like a nice kind of a throw rug or maybe like a table runner that you might want to have, very colorful and nice. Not really all that descriptive. One of the main things we wanted to get from this particular analysis is to actually see if our controls were good controls for our non-control. And one way we did that is we looked at all the organisms that we saw that are mussel, from the mussel beds, those are in purple. All the organisms that we saw from the non-mussel bed, those are in gold. If this sort of kind of cloud formation overlaps really well, and you don't have pockets of yellow in one place off by its own and pockets of purple over in places by themselves, that means that these two things are relatively close to each other with respect to the organisms, and that then the influence that the mussels have would be on some little pockets of organisms kind of nested within there. All right? So we should have a good, the organisms available there should be very similar, because we collected some from here in the Mississippi River and some right there in the Mississippi River, actually near Fairport, not too far from Muscatine and Pool 16. Great mussel bed right here. Up to 1 million mussels per acre, so 1 million mussels per the size of a football field. Lots of mussels in this particular area. Okay, so intrasample species diversity appears to be lower when mussels are present. So that means that there are fewer organisms overall in sediments where mussels are present. And so that's reflected by this data here. Every time you see this box lower than the blue data, that means that there are fewer organisms overall, all right, when mussels are present. Various statistical ways you can do this, all right, but you go all the way across there. Every time you see the pink kind of salmon colors for mussels, lower. So they suppress microbial diversity overall, all right? But approximately 12% of the intersample, or what's known as the beta diversity, was attributable to mussels. So anywhere you see orange, those are no mussel samples. Anywhere you see blue, those are with mussel samples. So you can see a clustering. There's very clearly an effect that mussels have on some of the organisms in this overall community. And so to unpack that more fully, we used some other statistical approaches for that and found that this particular organism, which is an anaerobic sulfate reducer, is statistically higher in the presence of mussels. This organism here, this chloroflexi, is a denitrifying organism, was higher in the presence of mussels. A facultative anaerobe and an obligate heterotroph, this chlorribi species, also higher in mussels. This one's interesting right here. It's statistically significant, but it's the reverse. Mussels actually suppress this organism, and it turns out that it's one of these ammonia oxidizing archaea. Wait a minute, I thought the mussels were giving this organism food, ammonia, and would make it like go away, do its job faster, so why would there be less? And so this significantly reduced AOA in the presence of mussels could slow nitritation, and so you're like, mussels to the rescue? Maybe not. Wait a minute, it's kind of blowing our whole theory here. And what's missing from this list? Anammox. And so we went hunting for anammox specifically using another technique, and I'll flash forward ahead to the results. At a 3 centimeter depth with mussels, there's statistically more anammox than there is at 3 centimeters with no mussels. All right, so if you do find that right zone below mussels, you actually do provide a favorable environment for these anammox bacteria. Okay, so can the influence of native freshwater mussels on the end cycle be quantified? I think I said yes earlier, and I'll stick with that. How might anammox impact our nitrogen pollution legacy? Well, they're already doing their job, right? The question is, can we restore our natural systems in these end polluted regions so that anammox can make a bigger difference? That's really the question. And then how do mussels influence the nitrotation anammox process in natural environments? Well, it says mussels mobilize nitrate, not so good, while suppressing anaerobic oxidizing archaea, while increasing anammox abundance in specific zones. So the jury's still out a little bit on the overall impact of mussels on systems like this. And with that, I will take 30 seconds of questions so we can stay fiercely on time. Okay, yeah, dive in.
Until 1:40.
Even better. I thought I had to be done at 1:30. Well, let me tell you 5 minutes more. No. I'm happy I moved through it quickly so I could have some Q&A.
So you sequenced the microbial community in the sediments. Did you think about sequencing any sort of symbionts that are right on the mussel?
We didn't think of that, but some of our colleagues have done that. So we've cited their work in our publication that just came out. It's pretty fascinating. And what they did was they did mussel shells. If that made any sense to folks, can you just scrape some bacteria off mussels themselves and maybe compare it to rocks like nearby or something like that? And that's what essentially they did. And it's pretty fascinating. Very different sorts of bacteria want to essentially populate the surface of the mussel. But one of the other interesting thing about mussel shells, they're kind of like shark skin. They've got a very interesting microtopography that you can only see under a microscope that actually inhibits biofilm formation on the shell itself. It's very resistant to bacteria growing on the shell. And so where you do see, though, and by the way, these particular mussels are almost fully burrowed. They're not sitting on top of the sediment waiting for other things to glom onto them. They're burrowed, but even within that, though, if you pull them out, they come out kind of clean. Things don't really stick to them that well. But where you do find those mussels are near the in-current and ex-current siphons, or those bacteria, where you can, and so that's really kind of a fun place to culture. Other people that are a little meaner to mussels than I am will actually sacrifice them and go looking for the gut, biota to see what's in there. That isn't what I like to do, so I let other people do that. But mussels are very interesting in the way they cultivate different bacterial communities.
So just a follow-up question. You didn't see anammox sequences in your Illumina data, but you saw it in the qPCR data. Was that the same sample?
So yes. So fantastic question. So one of the what would the word be? One of the advantages of non-targeted 16S amplicon sequencing is you get a little picture of everything, right? You get to see what everything is there. But then you can miss some details for organisms that aren't very abundant. And so we just literally were kind of below the detection limit of anammox using that technique. Hypothesizing, though, that they were there because we already knew it had elevated ammonia and nitrite, which are substrates for anammox. Then we used a targeted approach where we had the specific primers for the anammox, 16S gene sequence, and we amplified just those organisms and then quantified those. Then we had the sensitivity we needed to be able to see the anammox. So same sample, 2 different techniques to try to find various organisms. Yeah, Ted.
Dr. Craig, besides this, you do a tremendous amount of innovative technology. So mussels, other aquatic organisms, what's the future trying to apply that to wastewater treatment? Be it lagoons, be it something less expensive for cities and such?
I would love to put mussels in a lagoon. I feel a little bit mean to them, though, that then we just, flush ourselves all over them. But actually, I like the Iowa State researchers here that actually use an algae, and I don't feel quite as I don't have quite as much sentiment for these plants rather than I do mussels when we want them to do work for us. Freshwater mussels would be okay in some of these lagoons. They could certainly eat that algae. You'd have to put them in there yourself. Mussels reproduce with a required fish host, if you didn't know that. So a gravid mussel, a pregnant mussel, will actually attract a host fish. And they're very specific species of fish that go with specific species of mussels. When a host fish gets nearby, she'll inject her eggs into the fish, and they'll get caught on the gills of the fish. And then when the juvenile mussels, called Glaucidia, get big enough and the fish has swam away, that's how mussels spread. Mussels don't have wings or feet, right? Well, they do kind of have a foot. They don't move very far. Then when they get big enough, they fall off the fish, and then that's how they spread. So if you wanted to then, perpetuate that in a wastewater system, you would have to do that. Colleagues at Stanford University are using them in urban stormwater settings, which I think is a little bit more favorable, right? I mean, you can choke them out and kill them, right? We're going to have ammonia levels that are acutely toxic in our wastewater lagoons. So it may not work there, but urban stormwater does work pretty well. And I think it's a neat approach, especially if you're willing to repopulate them over time yourself, because they're not probably going to, naturally reproduce. I don't know where to go next.
In your mesocosms, did you look for anammox there? And were they there?
No, we did not. So yeah, the mesocosms was meant, that was a study for a different day, a different reason, and we never looked. And I suspect they would have been there, but they're slow growers, and our system only had a little bit of sediment. We may have never gotten into the zone where it would have been anaerobic enough to have that perfect sweet spot for them. And we wouldn't necessarily have imported them in from the Iowa River water, and so they probably wouldn't have grown fast enough for me to ever have enough of them.
So it would be worth repeating your mesocosm with a different sediment to actually see whether you get more N2 and less NO3?
So I would love, we learned a lot from those experiments. No one had really done experiments at the kind of the scale we were doing it. We worked with a freshwater biologist up at USGS in La Crosse, Wisconsin, a mussel expert. And so she was kind of our, eyes and ears to, the history of, research done. And so ours was quite unique, and we learned some unique things, but then it also had some significant limitations for some of the stuff you're describing. So we had to go over kind of some bigger picture kind of things that people hadn't done on the spaces in between, like this size on the bench and then the river. We were kind of in the middle. I don't know if even if we repeated it, we could do it quite like that. And then the other thing is, anytime you bring mussels in the lab, all you're doing, if you have a great lab setup, is just, if you're doing it right, you're just, keeping them alive longer than others. They're all on their way to dying if you keep them too long. And so I like to do the studies for no more than a couple months, and then I return them to the river. Otherwise, you start doing it much longer than that, and they're not happy, and they die, and then I feel bad because they might have been 50 years old, and who am I to go grab a mussel and kill it? So
you can cycle several populations potentially to build up your bacteria.
That's right.
Got it.
Yeah, and so that's why we're in the river now. That's why let's just go to the river where the mussels are, and I'll invade their homes and not collect them. So, yeah.
Did you look at T and QC ratios, carbon ratios, to see how the succession of the microbial population might change?
So no, but Teresa Newton, who I was mentioning, our fisheries biologist, has done that on mussels and on the sediments. So no, we didn't do much with that, and I'd have to even think of what I would hypothesize that would happen. But no, we didn't do it. Yeah.
Thank you.
Thanks all. Next speaker, right? Hey.
Yes.
Oh, you got a question.
But I did have a question.
All right.
So you put your model up there of muscle density and food /
Yeah.